Well, welcome to the second session of the NAI 20th Anniversary, A Discipline Reaches Maturity session. And we have a little bit more time this morning. You may notice that at the end of the session, there appears to be some free time in the program, but we are going to have lightning talks by the poster presenters. And so we will have some additional short talks at the end. And I think we do have time for 1 or 2 minutes of questions per paper. We'll stick with the 10-minute presentations with a warning at 8 minutes. So let's get started with Natalie Cabral.
Thank you, Carl. Great pleasure to be here this afternoon, and my perspective is going to be a very personal perspective on the NAI and what it did for me, but maybe as a case study of what it did for a number of young scientists. The getting started thing is really about the NAI when it started, but it's also me becoming a principal investigator exactly the same year at the SETI Institute. So what that says, exactly like Tori said this morning, I've never known anything else in my intellectual life as a scientist, you know, as the NAI. And definitely that has shaped my mind, the way I'm thinking as a young scientist, but now as still a very young scientist. I dye my hair gray. And so I would like to, get you through this because it's a personal journey, but I think it talks a lot about what is happening to young scientists within the NAI. So getting started, I came to the NAI, I was invited by Chris Chaiba for the first SETI Institute team. And I came from the standpoint of being involved with the Mars Exploration Rover team and thinking ahead, what is the next step? We demonstrated that there is habitability on the site on Mars. So if life was there, how does it look like? And the NAI seemed to be the perfect avenue to get into a multidisciplinary team and ask those questions and go to those places to start to figure out those things. And so I was a co-I on Chris's team, and with my 4 people strong task, and I think something like 75K, we decided to tackle these questions. But I think that what I did, especially at the very beginning, was more to test the system and the patience of a number of program directors, because I came to them and saying, well, there is this very, very big volcano in the Andes, and it's a really good analog to Mars. It's about 6,000 meters high. I want to go there. So they started to raise eyebrows, say, okay, and say, I made a reconnaissance over there. There is this lake inside that volcano. Oh, by the way, the volcano is not exactly extinct. And I'd like to dive in there. So you can thank me today if there is a safety review panel for extreme environment, because this is how it got started. Basically, even before the NAI with the DDF in 2002. So I tested that part of the system, but I thank NASA for that, because today we can make safe science in very extreme environment thanks to that. The passion of some program directors, because of administration in South America and sometimes boundaries that are a little flexible. I had a few phone calls from Mike Meyer calling me and saying, Natalie, I signed the paperwork for Bolivia. What were you doing in Chile? I said, I'm sorry, I was diving in that lake, and it's not my fault if the boundary runs across. But nobody asked me for my passport in the lake. So, but all what was fixed. But the point is that I was within a team, and I could put my samples into perspective. I was not on my own. I was not, especially when we arrived, I said that will be the fastest project ever, because the environment is going to be so nasty, we're not going to find anything. My task was put UV dosimeters over there, take the data, see what happens. And when we arrived over there, the first morning I realized I was sitting in a 100 square kilometer field of stromatolites. They were Pleistocene one, and then there were more growing in the current lake. So immediately the team in itself started to talk. They wanted more samples, and it's not necessarily the people in the team, but also the people they knew. And this started a network as a scientist for me. And then many of those people I'm still working with today. So we started, as I said, with little money for people. The project 5 years later was 50 scientists involved from 26 institutions, 9 countries. And we had reached out thousands of children in schools, including some events with the Exploratorium that were facilitated by the NAI in many ways. We also had, thanks to the DDF and other things, the Lewis and Clark funds as well. We had students every single year with us in the field. So always helpful and help get the project bigger. Also, the name of the NAI has opened many doors for me as a scientist. And even when the administration was very difficult, sometime in South America, you would be surprised that it seemed that people make a difference between NASA and NAI. And when we're talking about the NAI, actually it facilitated the paperwork. So for me, the NAI became a professional family and also a place where I could network, once again, with people I'm still working today. And also, it's a holistic approach to science. I am a firm believer that we go through science, like in life and respiration, through moments where you have to go and take the reductionist approach and look at the tiny window of your science, and sometimes you have to step back and look at the bigger perspective. And this is what NAI did for me, put everything into bigger perspective, and especially keep me in check intellectually. I remember at the very beginning of my career, I was writing lots of papers. And I was joking with my mother a few months ago because I was whining I could not write a paragraph, and she was very surprised to say, well, you seem to have more difficulty now. I said, no, I just know what I'm talking about now. And also the societal impact of the work that we were doing that probably would never have happened if I was not with the NAI. Going to those places, having high UV environment, finding life that resists to those things. All of a sudden, the oncology department at UCSC got involved with us, and they're still working with us so many years later because this might provide avenues for cancer research. So this is how this happened. It started there in 2003, and then I made the conscious decision to leave the NAI while SETI was trying to renew, and go to the more reductionist approach. So this is what I learned with the NAI. Now I'm going to try to apply it to mission, because this is what I do. I really want to apply that knowledge to mission. So I went through a number of P-stars and deployed rovers in the field, and tried to write a NAI proposal in 2012 that was not selected. So I went back to P-star, and Lakes on Titan had been discovered. So I applied some of that knowledge to missions for Titans. But then in 2015, there was the changing planetary environment and the Fingerprints of Light project. We started a new SETI Institute team. We started with 20 scientists. We're about double today. There was a constant. I heard the representative of CAP this morning talking about their involvement with the NAI. Well, Victor, you are here, and you know for a fact that we have been working together for 15 years. That started there too, and that continue on to with P-stars. So right now what we are doing with our project is to come in support of Mars mission and to bring the astrobiology in the biosignature exploration of the Mars 2020 mission, and think about how we can use the payload that they have to actually explore for biosignature. We are developing mission and mission concept. I'll talk about that tomorrow. And also the teaming with the IC World NAI team has led to a winning P-star proposal for the exploration of Europa. So we talk, we mix, we match, and we come from our different perspective. Also, what I appreciated very much is the possibility of giving my opinion on how I think astrobiology should move forward and have an impact at the Academy of Science. For now, in terms of research, what comes next is this application. How do I transfer the knowledge that we are acquiring with the NAI project into mission? And this is what I'm planning to do in the coming years, one way or another. This is my task, and we'll see how that happens, probably going back to a more mission-oriented view of things before we go back to multidisciplinary project. We'll see that I have no clear vision yet. But something that I want to conclude on is that at the same time that I became the PI of the SETI Institute team, the same year, abundance of goods, I became also the head of science of the SETI Institute. And it happens that we have 6 research divisions going across pretty much the program that you see at NASA. And definitely the way that the NAI has shaped my mind and have me think over the years is very much applied to the SETI Institute, where we are creating bridges among disciplines and see how, what are the new ways we can do more science, better science. So I'm going to leave that here. What I wanted to say is that, of course, we can, look back at the science, but it will never happen without the people who first created the NAI. But I have a special soft spot in my heart for the NAI Central. You guys are every single day, day in, day out, making it happen. And you'll go really, the extra mile to help us. So thank you very much, very, very much for everything.
We do have time for perhaps one quick question for Natalie, if anyone has one. Then thank you very much, Natalie. Okay, let us move on to Laurie Barge.
All right. Hi there. My name is Laurie Barge, and I am an investigation lead for the NAI Icy Worlds team at JPL. And I'm here representing the whole team, but most of all our PI, Ishik Kanik. He is on sabbatical right now in Italy. And so partly I'm going to talk about how our team has really benefited from the NAI's support and in terms of science and the whole team at JPL, but also Carl had asked me to speak about some of the early career benefits that I personally have experienced. So we'll have a little bit of both of that. And so our RC Worlds team has actually been part of the NAI for 2 separate cycles, that we were part of the CAN 5 and also part of the CAN 7. And so the CAN 5 started in, 2009. And so I was actually part of the team as a postdoc in the CAN 5. And then we wrote the CAN 7 proposal, and I was in that one as a co-I. And then when that got funded, it was a large part of helping me get hired at JPL. So that was one of the major advances of these types of teams, is that you can sort of be in them as an early career person for a long period of time, enough to sort of grow up and be able to contribute as a co-I, and then you can write the next versions of these things, and actually this will help a lot for early career people to become established at their institutions. And so the focus of the team evolved and changed a little bit over the 10 years, and in the CAN 7 team, which I am most familiar with, we were looking at, in general, the icy worlds, which are now called the ocean worlds, and we were interested in how you could determine whether these worlds that have the ice shell with the ocean, with the rocky seafloor beneath, how you could determine if those are habitable, and how you might approach looking for life on these worlds. And so one of the big questions that we focused on as part of this was not just understanding the moons themselves and how to detect stuff, but also trying to understand how life can begin on these worlds in the first place. Because it's possible that you have things that are habitable, but where life does not exist because it never emerged, right? And so one of the nice things about our team in the CAN 7 round was that we had this big origin of life contingent that was also working together very well with the, studying the interiors of the icy worlds and finding out what types of environments exist, but then also understanding how life could begin, how it could evolve and change, and how that might be represented in these worlds in a way that you could detect from the surface. And so we had 4 investigations. Investigation 1 was about looking at how water and rock interact and the chemistry that happens there. Investigation 2, which was the one that I was leading, is how geoelectrochemical systems can lead to the origin of life. So this was a hydrothermal origin of life work, which is for an ocean world, if there's no land, this is one of the main ways that you may have origin of life on these planets. Investigation 3 was looking at the structure of these moons and where the disequilibria exist, and Investigation 4 was looking about how we can understand the surface signatures that might come on the surface based on the stuff that's happening below in the ocean. And so there were lots and lots of different science accomplishments over the years from all of our co-Is. We had a large team that was spread not just at JPL, but also at outside institutions. We had international collaborators and lots of different involvement with other NAI teams as well. And so, not to go into any particular research project, but there were a lot of things that could come out of the fact that there was meaningful and long-term support. And so a lot of the stuff that we were publishing was things that had taken several years to sort of germinate. And being able to do field work multiple seasons in a row, or being able to do lab experiments that go on for 2 or 3 years before you can figure out a good method and this sort of thing, it was a really valuable way to be able to make high-impact contributions to the field. And so one of the things that was very important for me as an early career member of NAI, even before I joined Icy Worlds actually, was all the programs that NAI supported. So this ranges from, there were the AbGradCons, the Astrobiology graduate conferences, which I think many of you have also attended. Actually, many people who are now PIs or co-Is in the NAI have been attending these AbGradCons as well. And it was a graduate student-only conference, also postdocs could attend. But it was a conference where you could give like your first conference talk and get to make new collaborations with people. And so I attended AbGradCon actually, I think 2005 onward. And so I was a PhD student in Earth science, but I didn't have a planetary science or astrobiology thesis. But I wanted to be an astrobiologist. So I would attend the AbGradCon as a way to build the professional network that I wanted that I didn't really have from my institution. So this was really great. And then there was also all these astrobiology summer and winter schools. So I had to go, for these two I could find them online. This is the Santander Summer School in 2017 and 2018. For 2006, which was the one that I attended, I had to actually pull out a physical photo and scan this because I couldn't find it online. So this 2006 school is where I met a lot of the collaborators who I still work with today. And then this kind of continued throughout there was a Hawaii summer or a Hawaii winter school, an Iceland summer school. There was actually a University of Arizona winter school, as I recall, as well. And so I went to all of these, and you would see these people that were kind of building their own early career network that was transcending what they might be doing in their particular thesis program that allowed them to build a career in astrobiology. And so that is actually what led to me being involved with Icy Worlds. I was at AbGradCon, I think, in 2008 or 7, and I met somebody who knew Ishik, and they introduced me, and one thing led to another, and then I was a postdoc at JPL. So the AbGradCon was also a pathway for people to get into the teams and actually become then later co-Is. And another thing that was really beneficial for the early careers was the NAI Director's Discretionary Fund. So this was a small grant that you could apply for where it would be a couple tens of thousands of dollars to do a research project with another team or to go visit some other collaborator maybe that wasn't yet in the NAI. And so that was actually a really valuable way for a younger person on the team to sort of have their own idea and get funding to do it. And it's something that may not have been included in the original proposal. And so I had one of these along with Pablo Sobran from the SETI team, and this was a really big deal for us because we got to go to ELSI, which is the Earth Life Science Institute in Tokyo, and we were developing a laser spectroscopy project along with ELSI researchers. And we also had a student exchange where we had an undergrad come to Japan, and then later on, someone from Japan visited our lab, and then this actually, over a couple years later, it led to Pablo and I getting funded by a Peace Star, which I think Natalie just mentioned. And so we were able to get a project going and start doing preliminary stuff and start gathering team and thinking about things so that we could learn how to apply for these much larger programs. And so this was a really valuable thing for us to be able to work together. And a lot of that also came out of the AbGradCons as well. Another thing that was very valuable is kind of a smaller version of this. It was the NAI Early Career Collaboration Award. And this was about, I think, $3,000 to $5,000 where you could get this and then go visit another lab. And so this was where you could do a little pilot study or try out a new experiment or something. And I had one of these in 2012 where Bradley Burkhar from the RPI team and I wrote up a project to test RNA in a hydrothermal vent. And so I visited RPI for 2 or 3 weeks. We did a project, and then Bradley finished a lot of it, and then we had a paper in astrobiology that came from this. And it was the sort of collaboration that it would have been really hard to do otherwise because this isn't the sort of thing that will get funded right away. You need preliminary data. But also, it came directly out of AbGradCon again, because Bradley and I had met at AbGradCon, had this idea, wrote this thing with the support of our 2 PIs, and then ended up getting this collaboration going. And then later on, we ended up hosting several of these, and I think many of the teams have hosted early career researchers through this program. So in Icy Worlds recently, we hosted a student from University of Tulsa who came out to look at different ways we could analyze reactive minerals in early Earth systems. And then in 2018, we had another one come from Penn State to look at how we could have vesicles and compartmentalization in hydrothermal vents. So this has been really, really exciting to be able to have new people come and visit and actually get to work for several weeks in the lab, which is really a lot better than just chatting at a conference. But you need that preliminary data in order to move forward with things like the graduate student fellowship proposals, or even things like exobiology or habitable worlds. So the other one that was important for me in particular, this is a slide Ishik made kind of about the recent postdocs of our team, was the NPP program. So like many of the astrobiology programs, NAI could have NPPs with the teams. And you could also have postdocs that were affiliated with the teams funded just directly from the grant. And so I was an NAI MPP from 2013 to 2015, and before that I was a postdoc with IC Worlds for 2 more years, funded as a JPL postdoc. And so this was it was a really excellent opportunity to be able to have essentially my own fellowship money to do a project that was kind of a high risk, high reward type of project that wasn't, the sort of thing that you would get funded necessarily with one of the traditional programs. And throughout the years, we've had a lot of really excellent postdocs with the IC Worlds team. JPL has a very vigorous postdoc program, and so some of those individuals are shown here. And there's also others, I think, from the earlier can that I had not included in this slide. And then finally, we have so many excellent students that have benefited from the NAI. And at JPL, people often ask, so all your students must come from Caltech, right? And it's like, no, they come from all over, because you have these NASA internship programs where students can come from anywhere in the US or locally in California or even internationally, and they can work with our team for the summer, the spring, the fall, multiple semesters, anything goes. And so we have received support over these many years in order to really grow different students' careers and have them do research that became part of their thesis projects. They publish papers, they go to conferences. Here's an example of Ishik and I and 2 students were filming an article for the Journal of Visualized Experiments, which is a chemistry video journal, and Ishik did lab work and was in a small movie there. So we had a lot of excellent students doing great stuff with this team. And I think that is it. Oh, one more. Focus groups were a big deal too, and we had a lot of involvement with the TDE focus group, and this really enhanced our international collaborations in a big way, because we're able to have meetings at these institutes like ELSI and CAB and so on. And this was a major, major part of my networking throughout my postdoc years, and also has been hugely beneficial to our careers. Okay.
Thank you all. I think in the interest of time, we will move on to Frank Rosenzweig. There is a clicker if you want to use it. That's forward and that's back, but you can also do it right there.
Okay. All right. Good afternoon. Thank you very much for inviting me to speak at AbSciCon. It's always an honor to be here. So my point of departure is this: beyond the Darwinian threshold, major transitions viewed through the lens of astrobiology. And what I'd like to do in the next few minutes is pose and try to begin to answer these questions. What is the Darwinian threshold? What are major evolutionary transitions? Why should astrobiologists care about either? And how has the NAI advanced our understanding of these processes? So both concepts originate with 2 of the most original thinkers in the history of science, namely Darwin, arguably the preeminent biologist of the 19th century, and Carl Woese, arguably the preeminent biologist of the 20th century. And of course, Darwin's revolutionary idea is that all of life can be explained by natural processes. And his other revolutionary idea is that these relationships amongst all organisms, extant and extinct, can be explained in terms of ancestor-descendant relationships. Now, just as Darwin was an astute observer, an astute natural historian of visible forms, namely morphology, Woese was an astute natural historian of the invisible forms in biochemistry. And his revolutionary ideas were that you could study universal biomolecules, and in particular ribosomal RNAs, and establish these ancestor-descendant relationships and not only establish them, but provide evidence, conclusive evidence, that all of life is descended from a common ancestor. So in this cartoon from a paper by Eugene Koonin, he has another interesting idea, and that is the notion of genetic temperature, and that genetic temperature represents how information flows amongst replicons, whether those are populations of molecules or populations of organisms. And Woese made the distinction between information flow before the origin of cellular life, where you had free flow of information, and a constraint on that information above the point where cellular life arose. And this is what he refers to as the Darwinian threshold. So taking stock of astrobiology's traditional themes of prebiotic chemistry, of origin of life, life in extreme environments, and biosignatures, a group of us in the evolution community decided that perhaps there was an unfilled niche in the astrobiology portfolio, and that is the post-cellular evolution of complexity, and specifically major evolutionary transitions, these increases in complexity that followed the advent of the Darwinian threshold. So these major transitions can be viewed as transitions amongst from simple units to more complex wholes, and the assumption over time of interdependence and autonomy amongst those more complex wholes, which opens the possibility for division of labor. And of course, as this relationship becomes enforced amongst these subunits, the levels of selection change from this individual to this new individual. And so this is the, clearly, while we think of a lot of the tree of life and natural selection as occurring by competitive interactions, it's clear that many of life's major transitions involve cooperative ones, including the origin of metabolism, organelles, the various symbioses across the tree of life, as well as the advent of multicellularity. So all of these are involved microorganisms, and as such, these processes are open to the study to study using microorganisms and doing experimental evolution, where you can control the type and the intensity of selection, population size, and structure. And in Stephen Jay Gould's nice metaphor, it gives us the possibility of replaying the tape of life's history and examining the ecological and genetic forces that led, in these cases, to major evolutionary transitions. And so thus was born the CAN7 team Reliving the Past, Experimental Evolution of Major Transitions, and it's been my honor over the last 4 years to help coordinate the research efforts of this team of young investigators. And so some of the contributions that we've made over the last few years, we're interested in evolution of metabolism and how new genes and new protein activities arise. And here are some of the recent papers by some of our team members. And in particular, I would call attention to efforts by Shelley Copley and her associates, including Vaughn Cooper, who have demonstrated that enzyme promiscuity, which is an intrinsic property of all enzymes, is one of the major substrates for evolutionary change. So in terms of evolution of symbioses, how two or more species come to behave as one, John McCutcheon has established himself as perhaps the preeminent young investigator walking the earth today. Papers showing that the lichen symbiosis, which has been viewed as a simple two-member partnership for 150 years, is actually genomically much more complex. A recent paper in Nature showing that algal symbioses with corals are also more genomically complex. And of course, the elegant work showing that insect endosymbioses are these wonderful Matryoshka doll-like cell within cell within cell, genome within genome within genome organizations. And of course, we're interested in the evolution of multicellularity and how you get unicellular forms evolving from multicellular forms. And in this research, a couple of people, Matt Herron, a former NPP fellow and an NAI co-I, and Will Ratcliffe, also an NAI co-I, have made great strides. And so I'll give you just a little peek into some of their recent discoveries. Well, first of all, multicellularity has arisen many times in the history of life, at least a couple of dozen times, and this is within the eukarya, but it's arisen also amongst prokaryotes. How can we study the evolution of multicellularity? Well, we can study the fossil record, but the fossil record is relatively scant with respect to the early major transition to multicellularity. We can compare multicellular forms to their extant unicellular relatives, but of course there's a problem there in that each of these that these two forms have common ancestors, but they've been pursuing their own evolutionary trajectories for millions of years. For those groups where you have within a clade intermediate forms, such as you have within the chlorophyte algae, within the volvocene algal clade, you have the possibility of, you have single cell forms, you have transitional forms, you have fully formed differentiated multicellularity with germline and soma. And so what you can do in these cases then is to take this group, organize their cell biology in relationship to their phylogeny, and generate, in this case by Kirk in 2005, a 12-step program in relationship to the evolution of multicellularity. Or you can try to evolve in the lab a multicellular form that looks like one that's becoming multicellular. And this is what Matt and Will have done in a paper that was carried out in collaboration with Mike Travisano back in 2013. They showed that you could select for larger size just by increasing their settling through centrifugation and come up with a multicellular form that has its own life history in a single cell dispersal phase. And more recently, they've published a paper in Scientific Reports showing that you can do continuous selection in the presence of a unicellular predator, paramecium, and come up with these multicellular forms where you have a primitive control of cell number within the multicellular cluster. So this is what you got out of the predation experiments. This is what you get out of the predation experiments on top here. And while I'm not suggesting that they have evolved new species in the laboratory, it is true that these forms that they got in experimental evolution are quite reminiscent of actual bona fide species in nature. So you have differentiated multicellularity that can be evolved in the laboratory. These arise clonally and are therefore new units of selection. And the genetic bases of these de novo origins of multicellularity are now within our grasp. So in terms of broader impacts, the culmination of this work, and perhaps the influence of this work, was a recent conference at Georgia Tech, which was organized by Peter Conlon and NPP fellows at Georgia Tech, as well as Will Ratcliffe and Chris Reinhardt, who are NAI co-Is. And I would expect that going forward, many of the people who came from all over the world to this conference will be submitting proposals that populate the new RCN, the RCN from early cells to multicellularity. And so with that, I thank the NASA Astrobiology Institute, and I thank it for its funding for this high-risk, high-payoff research that has enabled us to attract a lot of other funding. Thank you very much.
Thank you very much, Frank. There's something that I had meant to say at the end, actually, of Laurie's talk, so I'm just going to go back a little bit. But your very last slide showing your leadership of the TDE focus group, and I always forget what TDE stands for, but the T is thermodynamics and disequilibrium, and entropy may be in there as well. But it was just a wonderful example of somebody who basically had their career development occur within the astrobiology ecosystem, if you like, then becoming an international leader because this focus group met internationally on a variety of continents and Laurie's developed collaborations really across the globe, particularly with folks at ELSI. So just, I think, a really wonderful example of how somebody can both benefit and then really contribute, not just scientifically, but to the community development as well. So with that, I would like to turn the podium over to Lauren Williams. Thank you.
Thank you, Carl. Thank you guys very much for inviting me here. Thanks for coming to hear me.
You can either advance there or your green is forward, red is back.
This will confuse me probably.
Okay.
Okay. Good job, Frank. I'm going to talk a little bit about, I've had to overcome some barriers to becoming an astrobiologist. I never met Carl Sagan, and I never held moon rocks in my hand. And I was trained as a biochemist, actually a biophysicist, and biophysicists don't think about time. You know, we study hemoglobin, we teach about hemoglobin as if it always was, and as if everything has it, and myosin and things like that. So that's the world I come from. But I was able to overcome this, and I made a really important contribution to astrobiology, which is I gave the Heimlich maneuver to Bill Schopf. And I'm personally, a lot of people have questioned my judgment on that, but I did it. It was just an impulse. And so I do have that. I put on my resume. Okay, so I started as a biochemist, and I had a long career studying ions, and I really thought I knew more than anyone in the world about how ions interact with nucleic acids. I studied the ribosome, but I also studied DNA bending, RNA folding. I had a long career studying ions. And then the ribosome structures came out, and I looked at them as databases so you could study ions even better. That was what I saw in the ribosome, was a lot of ions. And a couple things happened to me that sort of changed my life. Number one is I have a very good friend in Nicholas Hudd, who's probably not here because he never comes to my talks. Oh, wait, okay. Okay. Oh, I got burned on that. Okay. But anyway, Nick was my friend for a lot of years, and we have a lot of scientific overlap. Amazing kind of ways we look at things, but he has been very interested in the origin of life for a long time. And we talked about it a lot, and he kind of pulled me in, and he talked to me about this NAI and things like that, and kind of educated me. That was one of the things that happened. The other thing that happened is that one time I had ACL surgery, and I was lying on my couch. I had a long time to lie on my couch, and I was just like looking at the ribosome and looking at the ions in the ribosome, and just like, you know, I would just spend like days and days looking at these ions. And all of a sudden, I think, you know, I was also taking some narcotics at the time because of my surgery. But anyway, I thought, these ions have been here a long time. Yeah, and I just got this idea that the ions in the ribosome might tell us a story. And so and Nick encouraged me. I wrote up some ideas I had on how to think about this and submitted an NAI proposal, a CAN. And I really was an outsider. I didn't know. I'd never met Carl. I'd never met Ed or anybody in the NAI. And so I remember I was walking across campus, and my knee was still really bad, and I had this big box, and at one point I just said, I cannot get to the FedEx office, and I'm not, that was in the old days. It was a big box, right? It was a heavy box. It's not this electronic thing, right? You had to submit like 20 copies of a 200-page proposal, and it weighed a lot, and my knee hurt, and I called my wife. I said, I'm not submitting this proposal. And she just said, stop being a baby, and she came and got me and drove me to the FedEx, so I submitted it. So then I forgot about it. And I went on with my life, and a long time later, I was sitting in my house, and my phone rang. And this guy says, hi, I'm Carl Pilcher from the NAI, and we've awarded you this CAN, this center, NAI center. And my wife was right there, and I said, remember that I think I got that. And she said, no, you didn't. And in fact, she said, that's Nick playing a joke on you. That's what she said. She really said that. And then I said, it doesn't sound like Nick, And so, and then Carl's talking to me, and she starts Googling, and she shows me the webpage where NASA had announced it. And she said, you got it, right? So then she believed me. And Carl said, can you come up to Washington, D.C. Tomorrow for an NAI meeting? And I was like, yes, I'm coming. And so that's when I went up and I met Ed and Estelle and Melissa, and I met all the NAI staff, and I learned that I needed to know the moons of Saturn and Jupiter. I learned I needed to know a lot of things that I did not know. So I went up there, and I'd been studying these ions, and so then I think in 2009 there was this meeting in Phoenix, sort of a strategy meeting for the NAI, and I went to that meeting and I gave a talk on ions in the ribosome and what I thought I could learn about ions in the ribosome. And I was talking about magnesium, and the magnesiums in the ribosome are really remarkable. Okay, so this is a bacterial and archaeal ribosome. Okay, so this is going back before LUCA. These are magnesium ions that have been sitting here without moving for about 4 billion years. Okay, so this is just incredible. And they're hydrated, there's waters around them, and those waters have not moved. In fact, the protons on the waters have not moved. So this is like, this is the most permanent thing in the universe that is not cold, I think. Maybe somebody can argue with me, but I think that's true. Anyway, so I was talking about these magnesium ions and all the things we could learn about evolution, deep evolution. And I considered myself an expert. I'd been studying ions and nucleic acids for 20 years. I'd done a lot of crystallography, all sorts of stuff. And after my talk, this guy comes up to me. And I didn't know this guy. I didn't know anybody, actually, at that point. And he said, I think you're wrong about magnesium. He said, those magnesium ions are probably replacements of a previous ion, and they were originally iron. And I scoffed at him. I said, iron degrades RNA. You cannot have iron and RNA together. And I felt a little smug. And he said, you know the ancient Earth was anoxic, don't you? And I said, yes, even though I'm not sure. I can't remember if I really did. I may not have. I may not have known. Now in my mind, I think I did, but I'm not really sure. So that guy, of course, I should have known who he was, but anyway, I thought about what he said for a long time. And this was not really part of our NAI project or center to look at iron, but I just kept thinking, it was kind of haunting me, and really for, I think, maybe a year. And actually, we submitted, Lori talked about this, this little director's grant, and we submitted a little proposal. 2 minutes, okay, I gotta go fast. And Carl sent it out for review, and it was horribly reviewed, that we were gonna look at RNA and iron. And it came back, and he was like, Lauren, I wanna give this to you, but I can't, because people don't like your proposal. So we didn't get it, but ultimately, we did start working on this, and in fact, we wrote a paper where we showed that RNA folds beautifully with iron, and RNA works really well. In fact, and we submitted, and we had a very hard time publishing this paper. Okay, if you look, it's in PLOS. This is like the 5th journal we submitted it to. It was rejected everywhere, and finally, we sort of established this whole thing. And I'll just say that actually, there's Marcus has a presentation at 2:45 on Thursday where he's talking about our iron project. We have an ongoing iron project. It turns out we were right. They should have accepted our paper. And I just want to show you, this is our ancient Earth. I think we have the only dedicated facility in the world where you can study RNA and iron in the absence of oxygen. So this is our glove box devoted to iron in the absence of oxygen and the presence of iron. And that, I should say, that's Jennifer Glass over there, and she is my close collaborator on this project. And in the very back is Marcus, and he's talking about, basically, we can do translation. All the enzymes involved in translation, the ribosome, it works with iron. We can take the ribosome now and put it back to its ancient roots, its anoxic roots, and make it work in the presence of iron. So all of that is a story. I'd like to thank Carl. He just raised up the science I could do. And he just, such a good guy. And I'd like to thank the NAI and all the, Estelle and all the NAI staff, Melissa, everybody else. Thank you.
I love that story of Lauren's, not because I deserve any personal credit for it, but because we had a process in place that brought together investigators who didn't know each other, who had never worked together, and who would never have been talking to each other. And Lauren and Bob Hazen were two such investigators, and they came together at a meeting that we had organized for that purpose. And this is what emerged, a whole new understanding of the nature of the ribosome. So thank you very much, Lauren, for being there and for contributing that. And now I'd like to call up Jen Eigenbrode for a personal perspective.
There you go. So, Carl, when you emailed me to invite me to give this, I couldn't believe 20 years had gone by. And still, I'm having a bit trouble grasping all of that. I am a project scientist for two mission concepts. Both of them are searching for evidence of life on other places in the solar system. More importantly, 20-some years ago, I was one of the first PhD students that came out of the NAI program, out of an NAI program, meaning the first wave of us. It was astrobiology. It was NASA Astrobiology Institute that got me to where I am today. And so the perspective that I want to share is what that path was as a representation of all the richness that we have gotten from this and what NAI really means to the student side of those who have benefited the most, I think, from all of this, because what we have gained from NAI is still happening. It's still going to affect the future and the next generations. So before I get started really on all those details, yesterday 3 terms came up, and I was kind of shocked that I never really reflected on what they meant. There's multidisciplinary. So here's one of my favorite paintings by Rothko. It's, he just likes to put in different splotches of color. And these, to me, represent the 2 different disciplines. They're put together and they work together. It's just a representation. But this is interdisciplinary. And imagine all of these colors up here, all these little spots being different disciplines, and they merge together. And when they merge together, you can do different things. And so, for instance, biology, chemistry, geology, physics, start bringing in planetary stuff. All of that stuff starts to blend together. And what you end up with is something bigger. It's a bigger picture. And it's these bigger pictures that we couldn't really address until we had the tools and the programs and the structures, the infrastructure, to help make this science more or less transdisciplinary. And what we mean by that is all of these disciplines integrating together in such a way that they become more or less holistic. It's a single strategy going forward, but they're all blended together. We drop the walls. So let's go back to me, where I started. This is me up in British Columbia. I love the wilderness. I was in one of the last of the old school geology field camps. We're out there mapping geology in Tagish Lake area. And so I'm over here on the side here. And I was a hardcore geologist. I mean, this is the things I did. I even went into geophysics for a little while. I went to Bird Station, flew around for 2 months over the ice looking at whether or not there were volcanoes under the ice, where the ice flowing, all these cool, interesting things. But you know what? Geophysics just wasn't quite my thing. So I moved on, and I studied paleoclimatology. Paleoclimatology to me is an example of this multidisciplinary approach. At least at that time it was. I did chemistry and I did geology, some mineralogy in there. kind of brought those things together, trying to understand things. But there were things missing. It was like there were gaps in how everything connected, and it didn't make sense to me. There was something missing there. So that's when I went after what we would call Earth system science and trying to understand how everything gets connected. That took me to Penn State. So again, I'm still a geologist at this point. Here I am on the roadside with Lee Kump, over in the Book Cliffs, one of the classic places we look at stratigraphy, trying to understand all of this, trying to just really absorbing everything that's being thrown at me from the geology side, but still seeking the Earth system science side. And it was at this time that I got assimilated into NAI as a PhD student. And talk about opening doors. When we talk about, I'm not talking about my career, I'm talking about how I think about everything around me as a scientist, the questions that I ask. As a geologist, I was asking questions about biology. That seemed odd. There weren't a lot of other people doing it. And here's where a classic picture for my PhD type work. I went to the Hammersley Basin in Australia, investigated the geology that's there, the biogeochemistry. Here, I have on the bottom here, if you can barely see it, this was my, the category of discipline that I was in: paleo, isotope, organic, biogeochemistry, and stratigraphy. That is interdisciplinary. That is it. I mean, don't ask me to repeat it. and the craziest, on top of all that, when it came time to take my comprehensive exam for the PhD, Kate Freeman confided me, and she's like, well, we don't really know how to do this. There was only a few of us who had gotten to this point. So we're going to make you get tested on all of these topics. So I had analytical chemistry, isotope geochemistry, microbial biogeochemistry, and stratigraphy. And then one of those questions, I said this is an open book test, you had 2 hours to answer each question. One of those questions was, how would you do stratigraphy on Mars? How ironic it is that I'm working on Mars now, because at that time, we weren't sure if the river channels that were there were formed by water or maybe liquid CO2. We just didn't know. So things have come a long way since then. As I progressed through my career, so did the science and the breadth of that. So just to kind of spin through a few of these, here I am up in Svalbard, Murchison Fjord, trying to understand that paleo, isotope, organic biogeochemistry in a different place, but also the continuum of that record the whole way through today. What are the modern organisms that are there? What are they doing? And can we decipher that? We wanted to look at that life. What is it? Well, in extreme environments, it's hard to get to it when you've got so much contamination around you. We are walking clouds of microbiome. We alone contaminate things. So we spent 2 years trying to figure out how to clean things. This is a picture of Liana Benning swabbing the inside of a core barrel. And then when we finally figured out how to clean things, we actually went out to the glaciers and started looking for signatures of life. Okay, we want to do that on other ocean worlds now. Back then, when we did this, most of the stuff that we got was contaminated, still. And we didn't have techniques that worked very well. I mean, a lot of the approaches that we used, while transdisciplinary in nature and how we wanted to do it, the techniques themselves didn't work. So we're back to the lab. Here I am with Melissa Traynor. We're working in the lab together doing technology developments. How do we make the measurements we need on another planet? This is another shift. Okay, so here I am. The breadth of what I am doing as an interdisciplinary, transdisciplinary scientist is expanding. Meanwhile, all along, NASA Astrobiology Institute has been fundamental in making all of this stuff happen. In this case, it was Goddard Center of Astrobiology. Atacama Desert, okay, yeah, we finally figured out how to do things cleanly. And look at this. We've got a new technology. It's called a kitchen spoon. So this is how we were sampling in the Atacama Desert, looking for signs of extreme life that's living there. And we did indeed find that. But when we go to apply that kind of knowledge to a place like Mars, or other places in the solar system, we have to be cognizant of the things that we're not familiar with, like radiation. So here's a picture of a cyclotron where we were exposing analog materials to 8 million years worth of 200 mega electron volts of protons. That's a lot of radiation, but that's what you get on places like Mars, or en route from here to Mars, or something like that. So we were trying to understand, well, what happens to this type of stuff if it was ever there? Again, the transdisciplinary, the bubble, the cloud of all the stuff that we take in is growing. This is just my 2 cents on this, but there's a whole community that's working on this. And a lot of this culminates in something like a mission. Here is a picture of SAM. It's the gold box. It's going into the base of the Curiosity rover. And we've been on Mars now for over 6 years, exploring all the types of stuff that I was exploring as a geologist, as an organic biogeochemist, even isotope biogeochemist. All this stuff is going on. I'm a geologist again, but I'm also an astrobiologist through the eyes and tools of a rover. It's come full circle to some degree in that, I started off as a geologist, I ended up here doing Mars geology, but it carried with me all of the astrobiology with it. And now I've moved on to other places. I've gone back to the ice. Here's Enceladus. Enceladus is one of the places where I am a project scientist for a mission concept. So I want to kind of end this with just one of those nice little concluding remarks. When I was first asked to do this talk, I wasn't sure what I was going to say. But then reflecting after yesterday's conversation at the town hall on the multidisciplinary versus interdisciplinary versus transdisciplinary, it made me realize that NAI has really enabled me and generations of scientists to think broader than disciplines. Their disciplines can be like walls to some degree or buckets. And NAI has allowed us to not even see them anymore. I was talking to Alex Pavlov yesterday. He is an atmospheric modeler. And that's what his training is. And he's actually doing microbial simulations, like he's doing Mars simulations with microbes in the lab. He's doing biology now. All to feed back into his atmospheric studies, but it's another example of how we've kind of broken through what used to be walls. They're not disciplines anymore. NAI has really allowed the science of understanding life to evolve into something uniquely different than it ever was previously. And it's hard to explain what that is, but it's sorry, Mary, you're looking you're looking shocked. It's something uniquely different than what we had previously thought. I'm not saying life, I'm saying life as the search for how we think of it, how we study it. And in doing so, we really are turning the next chapter in astrobiology and searching for life beyond Earth. And I think that all of us here may actually get to see that search start up again and take place. And I'm sure we're all looking forward to it. So with that, I'll end.
Jen, thank you so much. When I sent that email to you, I knew you would come up with something perfect, as you did. Okay. Let me just say, while Karen Meach is coming up, Karen is the next speaker. I'd just like to say that it's really wonderful to have a few representative stories, such as Laurie's, such as Jen's. We're going to hear at least one more today. But it's just been so gratifying over the years to see so many young researchers come up through this ecosystem, and it's been one of the things that I feel really privileged to have been able to support. Karen? I'm forgetting to do my job. There we go.
Thank you very much for the invitation. So I want to give a perspective on the University of Hawaii Astrobiology Institute and some of the lessons learned. And I'd like to start with when we first proposed. There was actually a failed proposal, I think either Can 1 or Can 2, in part because it was too narrowly focused. So when Can 3 came around, we started to look at the existing teams and saw that many of them were multidisciplinary, covering a whole range of topics. And to me, that seemed like it's the wrong way to go. And I really liked the VPL model of a single focus question where you address it from many different disciplines. So our team came up with the idea of having a watery Drake equation, or exploring the formation of habitable worlds through the story of water, bringing in disciplines such as cosmochemistry, astronomy, geochemistry, disk chemistry, and even in the early years, then looking at extremophiles. But I'd like to start with some of the ways in which we fostered this interdisciplinarity and how it's resulted in some activities. Most of our funding, like many of the teams, went into funding the early career scientists, postdocs, and graduate students. And we decided that it was very important to have them co-located so they could talk to each other. And I think someone this morning said it's not just about putting people together. Well, sometimes it is. You have to learn each other's language. You have to understand what you don't know. And that's what happened by throwing our scientists together. They started to learn about other disciplines. This resulted in a lot of interdisciplinary collaborations that spread further with some of our senior scientists looking at how disciplines interact with each other, as you can see in the graphic in the lower right, and it spread to investigating how teams across NAI started to talk to each other. I think one of the big, really important fundamental programs that we had was the development of summer and winter astrobiology schools. This was a brainchild of Bo Ryperth. He said, you're never going to build a network of scientists unless they start to get to know each other. And the best way to do this is in a focused environment, not just where they're teaching them different sciences, but you have the opportunity to network, to learn about each other in social environments. And so this started the 2-week-long schools. We began to collaborate with Wolf Geppert at the Nordic Astrobiology Institute, and this has resulted in 135 alumni that I now see very proudly are leaders in the field of astrobiology. And I'd like to think this also helped play a role in the Europeans really ramping up, and they're now forming their own astrobiology institute. So I want to give you 2 examples of why stuffing the postdocs into the same office was useful. One day, James Stevenson, who was a bioinformaticist, worked in microbial environments, turned to Lydia Hallis, who's a cosmochemist working on Mars meteorites, and just asked, how much boron is there on Mars? And she said, why on earth would I care? And he said, well, it's kind of important for life. And they had a long discussion. She said, well, you know what? I've got meteorites in the lab from Mars, and I have ion probe time tomorrow. Why don't we measure something? And so they did, and it became a wonderful collaboration that led to a very high-profile paper looking at the possibility of formation of ribose in an environment conducive to life on Mars. Another area was tracing the origin of Earth's water. Many people for many years since the missions to comets, Halley's Comet, had thought about D to H, isotopes as a tracer of the origin of water on Earth, but everybody was looking at this from the point of view of comparing it to Earth-ocean water. And a bunch of the team started to get together and think, well, the geologists said there's more water inside the Earth than on the Earth. The astronomers said there's water in the Earth. And everyone started to think about it and said, you know what, maybe the water on the Earth isn't what we should be measuring. And so that led to geology expeditions, Baffin Island, Iceland, and the discovery that probably the water in the Earth is very different. And this has led to some remarkable questions about how we really would trace the origin of water in the solar system. So many years of work with the team has led to some ideas developing a mission. And many of you have seen these marvelous pictures from ALMA showing nowadays that we can actually look at the process of birth of planets and other star systems. You see these dark lanes of gas gas and dust, where maybe planets have cleared out gaps. We're looking at the process of planet formation, but what we're not seeing is the formation of planets in the habitable zone. What we really want to understand is how you build a world that has the right ingredients for life. People are proposing now for the decadal survey the next generation of these very large arrays, which will have the ability now, the resolution and the sensitivity to watch this process unfold in extrasolar planetary systems. We will watch the formation of planets in habitable zones and look at their chemistry. But the question is, how would you do this in our solar system? We can't go back in time, so what we have to do is look at the leftover remnants of the process here. Namely, we want to trace the isotopes in the young solar system, and that's best done with primitive objects that haven't been altered before. Cosmochemists have been looking at meteorites for years. They've been thermally altered. We've had spectacular measurements from comet missions. They're pristine, preserved remnants of the early solar system, but we don't really know the dynamical history of particular objects. We've explored icy satellites, which have thermally evolved, and I won't even bring up the interstellar objects because we only have one so far. However, we have icy asteroids that are preserving this primitive signature. And I think one lesson to be learned from bringing the disciplines together is this is not an easy question. You get a chemical fingerprint in all the building blocks that make up a solar system, and that chemical fingerprint depends on a gradient in temperature, it depends on the physics, and each isotope will behave in a different manner, so you have different tracers. But then it all gets scrambled up as the planets are assembling. So if we imagine that in our solar system, as the current paradigm now thinks, Jupiter formed early, probably opened a gap in the disk. This impedes the inward flow of icy material such that the habitable worlds in our solar system formed in the hot inner disk, where it's harder to get volatiles and organics. But then as Jupiter grows, its gaseous envelope gives it a lot of mass, and it starts to toss around the planetesimals. Where they come from depends on whether Jupiter formed and didn't move or whether it migrated. So the key, I think, is to actually sample the region where Jupiter tossed material, and that's in the outer asteroid belt. So in fact, by combining different disciplines, cosmochemistry, disk dynamics, interstellar medium chemistry, and Earth geology, you can begin to look at where did the material come from in our solar system that brings habitable ingredients or ingredients to a habitable world, and we can compare that to extrasolar systems. And so I think the way to do that is with a Discovery class mission. And so that's where combining the disciplines with our team has been leading. I just want to finish off with where it started. And I think, again, one of the really fundamental things, aside from allowing different disciplines to talk to each other with NAI, has been the remarkable ability to train the next generation and form those critical networks where you actually think outside the box, ask that stupid kindergartner question. I mean, I'm sure all of you have done outreach with kindergartners. They are not afraid to ask that stupid question. And it's remarkable when you get adults together. They're not afraid to ask the stupid question if it's not in their field because they're not supposed to know that answer. So by having these astrobiology summer and winter schools, we've given the people the environment and the conditions to start that discussion. And so now we have a generation of people ready to ask these questions across different disciplines. Thank you.
Thank you very much, Karen. Karen's work really has been so instrumental in that ecosystem that I've referred to of training and educating the next generation of astrobiologists. And now I turn the podium over to our co-convener, Dave DeMarais.
So before lunch, Carl said, now with Tori Holler's talk, we're transitioning into the afternoon where you're going to hear from early career investigators and mid-career. And I said, oh, God. But then actually, as I was eating lunch, I thought of Bill Shup's talk, and that my early career experience actually was with PPRG. And everything that Bill said about teamwork and having a good time and everything just totally applied. And in many ways, that was the sort of the forerunner spirit of what we're talking about today. So, this is the longer view, I guess, going back a few decades on that same topic. Microbial mats. Again, it was Bill that introduced me to going to Baja and seeing these wonderful mats down there. And the perspective in my life that changed was that I thought of biofilms as something that you brush off your teeth twice a day and an outcome of bad housekeeping in your bathroom. But actually, I found that there's a lot more to biofilms, as you will see shortly, and I'm sure you already know. So here's the connection here, and I guess to get to the details of it, there's just a number of reasons why these photosynthetic microbial mats are really important in astrobiology. I think they're one of the central nodes of what it is that we're doing. First of all, they're the oldest, most widespread ecosystems that we have documented so far. They're remarkably complete self-contained ecosystems, so it's everything you want to know just in your hand. They've probably hosted several key evolutionary events given their antiquity and their importance in ecology. And the key thing for physiology is that the microenvironments within mats differ markedly from the external environment. Steep changing chemical and light gradients mean that the physiology that's relevant is not something you measure in a culture medium in a pure solution, pure culture. And of course, the punchline again for understanding the early rock record is mats create abundant and diverse biosignatures. So there's all kinds of incentives for studying these things. This wiring diagram, which is sort of the ecosystem of the mat, starting, you push the back button, it goes back. Amazing. This is the button I want to push. This is basically an entire ecosystem, rather like a forest with the canopy organisms, the cyanobacteria, and the carbon flowing down through all these other organisms creates this incredibly diverse ecosystem. But in the theme of this session, what I really want to highlight is that this is a fundamentally interdisciplinary topic of study. And that is to say that you've obviously got the microbiology in here, but you have the chemistry because you have a lot of photochemistry that's actually happening with the chemical intermediates that are here. There's a lot of physics in this because we're talking about the light regime and how scattering and other optical effects really determine that microenvironment that is so important for shaping the physiologies of these things. And then, of course, you have the geologic aspect, and that is that the products that could potentially go into the geologic record are subject to a bunch of processes of great interest to sedimentologists and geologists. So, it's really an argument right here for it really takes a team, an ecosystem of scientists of diverse capabilities to study this kind of ecosystem. And the shaming aspect of it is that once you realize how exquisite the system is, you figure, Jesus, my team had better be at least half as efficient as this community is that we're trying to study. And so it does motivate you, given the incredible fine-tuning that has been done in this ecosystem over time. One key example of this is that the rates of oxygenic photosynthesis that we measured are the highest rates that have ever been measured on Earth, compared either to a coastal ecosystem or a rainforest. So these things are incredibly productive. Part of the reason for that, which we discovered by making these rate measurements, is that all the major processes within the mats, oxygen production, sulfate reduction, oxic respiration, and so forth, are all finely tuned to match to each other. And when you change the temperature, they all change in concert. If you took each of these processes independently, they would change differently as a function of temperature. You put them in the ecosystem, they are lockstep with each other. And this high coupling efficiency is part of the reason they can drive these very high rates of primary production. And to understand that, you have to understand the ecosystem. So that's just another example of how the whole being greater than the sum of the parts. One of the big benefits then of being involved with NAI, we had done this work for a number of years, but then NAI just allowed us to scale up big time to get a number of people to collaborate with and just to approach it at the proper scale to really begin to drill into this. A great collaboration with the University of Colorado. John Spear mentioned this, I think, earlier. This key paper by Ruth Lay. This was one of the first real demonstrations of Norm Pace's environmental sort of genomics capability. just an incredible diversity. In fact, we never could really converge on what the total number of so-called species were in these things, and just really looked into a big dark room with a very dim flashlight saying, oh man, there's more to these mats than we had figured. And it was through the NEI with this collaboration that we were able to make that revelation. Of course, now that's common. People have done this in a number of ecosystems. Yeah, of course, it's incredibly complex out there, but this is really one of the first times that we were able to demonstrate that. We took that forward looking into gypsum crusts later on. With NAI support, we were actually able to map the different types of phototrophic organisms down through the gypsum mat. These are, of course, the cyanobacteria and the diatoms at the top, and the purple photosynthetic bacteria, and basically anoxygenic phototrophs further down. And this is where we began to really couple the populations and their distribution with depth with the actual functions that were going on that we could measure with other methodologies. And really, this is now, I think, where all ecology is going right now. You have this ability to map these populations and understand their physiologies, but now in the context of how they work together in a physiologic sense within the ecosystem. And this was, again, a direct outcome of the ability to incorporate specialists who can do the genetic work, the people actually making the physiologic measurements. And of course, just a visually beautiful demonstration of this was when Victoria Orphan joined our group, again, during the time we had the NAI support. And she was she's a genius in a number of ways, but one of the ways is adapting staining techniques to some really challenging environments. And here you're looking at trying to do fluorescent tags in a community that just glows in the dark when you hit it with light because of all the photopigments that are in there. And yet she figured out a way to make probes for methanogens, actually, but also later on, working with David Fike, probes for sulfate-reducing bacteria. So when you look at, this is like a millimeter here, and you're looking down through the mat, you see the photopigments are glowing here in red. The DNA stain, of course, is all over the place. And so when you put those two together, you get purple. So you're looking at cyanobacteria here. But the thing that's amazing about this is the SRB, you have sulfate reducers right up in the oxygen-rich zone. And as you go deeper in the mat, you see different types of sulfate-reducing bacteria as indicated by the fluorescent tags. Now, one of the measurements we made early on was that sulfate reduction, which is an anaerobic process, is poisoned by oxygen. And yet we measured the highest rates of sulfate reduction ever measured in nature in the oxic zone of these mats. Now, if you want to get a slap across the face saying you really don't know what's going on in these things, just try understanding why an obligately anaerobic process is happening in the presence of 100% oxygen saturation. And we made that measurement earlier, and with Victoria working with David Fike, actually started putting some names and faces on the characters that are capable of doing that. And yet, as you go deeper within the mat, you're seeing different types of SRB, indicating, again, adaptations to different environments in this one group of bacteria. So we're really getting closer to understanding the machinery inside the engine room with these communities. And then, of course, as we go to biomarkers, and this is a slide that Nikki Peranto showed yesterday, done by Linda Janke, we ultimately want to get this complexity and diversity of this community and see how it maps into the geologic record. 2 minutes? Great. And as a result of that, Linda has, of course, been the real genius in this whole thing, is just really done catalogs and catalogs of different types of lipids, which types of organisms they map to. Just an incredible, detailed study of the literature followed by lab work to where we really now see an enormous diversity of lipids that these mats are generating. One of the real opportunities of these mats, and something that we haven't realized until recently, is that by looking at these modern examples of cyanobacterial mats and also studying ancient stromatolites, such as this one from the Sibley group in Ontario, we can begin to correlate the diagenetic fates of the fabrics of these stromatolites with the diagenetic fates of organic biosignatures, and how the lipids are actually preserved and into these stromatolites, and how there's a potentially great record there. One of the things we realize is that a lot of these beautiful ancient stromatolites must have lithified early when the organisms are still alive in order for those fabrics to be preserved. And this is just demonstrated by the fact that we have a time scale on these mats. We see how the fabrics are changing. And it's just a great Rosetta Stone for correlating two types of biosignatures, the fabric biosignature as well as the organic biosignatures. And so with that, I just want to mention, of course, that on Curiosity rover, we're driving up the hill here, and there's evaporites up ahead on the road. We're actually going to be able to look at sulfate deposits on Mars. And these two images just show that when you look at the scale of observation in Baja, a centimeter scale here, we're already at that relevant scale of observation on Mars. This happens to be Meridiani Planum here. Of course, the tip-off is that we haven't found the stromatolites yet, but the point is we are at the scale of observation where you could actually begin to seriously interrogate Mars at a scale relevant of observation. With that, I'd just like to thank for support, and I just want to make this general point that NASA and exobiology and astrobiology's support of team research goes way back actually before NAI, witness what Bill told us earlier. And that's the key thing, you know. Maybe the names of the organizations change over time, Institute maybe now transitioning to something else, but what's important is this long-term NASA commitment to team-oriented interdisciplinary research, and I think what we've seen today with these talks demonstrates the incredible payoff that comes from that. So thank you.
Thank you very much, Dave. Just one bit of housekeeping before I call up Chris, and that is, could I just have a show of hands so I can get a count of how many of our poster presenters are here in the room? Okay, there were more hands than we had poster presenters. So, okay, we will, because there were 6 posters associated with this session. Yeah, so could I have another show of hands of the people who had posters associated with this session who are in the room? So there's 1, 2, 3, and you would all like to, and so you're all going to be invited to come on up, and if you have illustrations on a stick, we should be able to accommodate it, and if you need to show it from your computer, we probably can accommodate even that, but a stick would be better and easier. And with that, Chris House.
All right, thank you. I want to thank Carl and NASA and Penny for this opportunity. Like many people who have talked before, there are some common themes. The careers diverted, opportunities to collaborate enabled, opportunities within one's own career to be enabled. So my story is slightly different in the essence that my career got diverted partly before NAI got started. So I wanted to start, you can basically see the bottom, all these acknowledgments is my roadmap, if you will. And the far left, I want to start with actually the NASA NSCORT program, which was a predecessor to NAI. There was a NASA NSCORT in exobiology at UC San Diego, and there was a NASA NSCORT in exobiology at RPI. And I was part of the UC San Diego one. And then I'll go through, like Jen Eigenbro did go through the career, but I won't talk about our comps, which I was also part of on the other side of that. So here I am as a sophomore, and this is me as a sophomore, not that picture. And I didn't go to UCSD for astrobiology. In fact, I'd never heard of that word. But I did know from my high school textbook who Stanley Miller was. And I very much idolized his work. Had no idea he was at UC San Diego. And one time, as actually at midnight, I called the captain of the UC San Diego club sailing team to tell them that I wasn't going to be able to do practices in the spring because I had to take OChem, and which was going to be in the afternoon. And she said, well, if you're going to miss the practices, you might as well take Stanley Miller's class. And I said, really? Okay. So the very next day, I went to Stanley Miller's office and knocked on his door, and he let me in. And I sat down, and this is my confession to everyone. There's been some confessions. I said, I really like to take your class in the spring, and I don't have the prerequisites. I'm not a chem major. I'm sort of maybe a bio major. I think I'm officially anthropology if you look at my, Anyway, well, he said, do you know what glycolysis is? I kind of exaggerated. And he said, okay, you can take the class. So I took his class, and then I joined his lab, and that was did summer research with the N-scored in exobiology, and it was fantastic. So that's the my career diverted into astrobiology. This is a lab meeting with Stanley. There's Stanley Miller. I'm in the middle there. Jason Dworkin is sitting on the lab bench, and so he's project scientist for
I've got a laser here if you want to use it. It's the little green button up there.
There's Jason Dworkin. And so he's project scientist of OSIRIS-REx, if you don't recognize the name. Tony Keefe, Mike Robertson, Pepita Loretta, and Michael Robinson. Sorry, shoot. Anyway, and then anyway, I'll think of it. So next, I went to grad school, and Bill Schaaf was kind enough to let me into his lab, and did a wonderful job training me. But this was prior to NAI, and so you've heard about all these summer schools you go to every single summer. You went to all 5 of them or whatever. No, that was not part of. I mean, there were a few summer schools. There was the Woods Hole Microbial Diversity one, goes way back, but there it was a small community. Astrobiology was a community, but it was a very small community of grad students. Mainly, it was the Gordon Research Conference, The Origin of Life, where we'd see each other. But then UCLA was a founding member of NAI in 1998, so I helped with that proposal, and we got funded. And then I graduated. By the way, this is the Grand Canyon. And I went to Penn State, and this is really, I think, the main, in terms of the history of NAI, this kind of celebration we're having. Our role has been to train a high number of excellent graduate students. And I think Penn State does attract excellent graduate students. Jen is a good example. So we've had 90 astrobiology-related PhDs. And in the interest of time, I decided not to go through every one of those dissertations today. But instead, just to say, that many of them are in the room, many of them are at this conference, and it went through basically. We had Can 1, Can 1 and Can 3. Hiroshi Emoto was PI. Can 5, I was PI, and then now Can 8, Kate Freeman is PI. So Penn State is back in NAI for I guess that's a 4th time. All right, so we train the next generation. That's largely what we did. If you look around AbSciCon, I currently run into about a dozen of our former students. It's probably more like 24. I just didn't run into them all. And one example that just to really illustrate it it's a really nice example, is Goddard. At NASA Goddard, you have 1, 2, 3, 4, 5, and across the street 6. 6 Penn State, former Penn State PhDs. There may be more. I may have missed them. I think they must have an alumni lunch they go to every day. I don't know. They should. But in terms of opportunities to collaborate has been another common theme. And for me, I think this example, I tried to think of an example that was really dramatic and totally changed my career. And that was as a assistant professor, having NAI membership and having NAI funding provided me the opportunity to meet and to then launch a project right out of the blue with then graduate student Victoria Orphan and then postdoc Kai Hendricks. So 3 very junior people at the time. We're not junior anymore. We've aged. But together we launched a productive research project on ANME cells in methane seeps. And that led to many trips out to sea with Victoria. Then that also led to IODP cruises. And that's another thing that NAI enabled, is that while the International Ocean Drilling Program provides a ship and provides sediment and provides an opportunity, you don't necessarily have a lot of research funding to do anything with that opportunity. And so having the funding in place and to do astrobiology out at sea, the combination is excellent. In terms of enabling too, I'll just say that this continues, and somewhere around 6 days from now, between now and 6 days, I will push the submit button on a NASA Discovery proposal. So it's really just going to be excellent. We'll see what happens. It's just a proposal, but it's a really great one. An astrobiology lander to Ceres. If you want to see that, that's tomorrow in e-lightning talk. Okay. So it's provided unique opportunities, especially for junior scientists. It's allowed for high-risk, high-reward science, and there's been a tremendous community created through the critical mass it's created, the interactiveness, the internationalness, and the collaborativeness of NAI and its related organizations. We are moving ahead, so we've gone from NSCORE to NAI to the future. And so I think I just want to end by saying that I'm optimistic that this will really be a similarly great program as NSCORE and NAI, and that it really will be what we make it, because it has a grassroots sort of flavor to it, that we can populate these nodes, and we can interact, and we can do it if we choose to. And then there'll still be the alternative that ICAR provides a great longer multi-co-I grant opportunity. So I think all the framework is there if the community accepts it and really runs with it. Thank you.
I just want to particularly thank Chris for telling his story. I've told Chris on more than one occasion that he was my poster child for illustrating the whole process, and I think it's an important part of that process that Chris began before the Origins program, which is what emerged from the ALH 84001 announcement in 1996. And that Origins program included the NAI, but it included many, many other things as well, because the astrobiology program, as it developed following ALH 84001, was in turn built on the foundation provided by decades of exobiology program funding to this community from NASA and the NSCOREs and PPRG and a variety of things. And so again, we continually stand on the shoulders of those and what came before, and we benefit from it and see farther and go farther because of it. So now we have time for, I think, 3 lightning talks, and we'll do it in the order that it was in the poster session. And so Sanjoy, would you like to come up? And we may have to do a little bit of shuffling. Do you have anything you want to show? Okay, in that case. I'm just going to return there. Okay.
Good afternoon.
You nominally have 5 minutes, but we've got half an hour to go, so.
Okay, I'll try to be quick.
You can be relaxed.
My name is Sanjay Som. Please come check out our poster number 17. The idea behind our poster is that during graduate school, I founded a nonprofit to build community around doing science. And we've been around for 10 years, and believe it or not, this is the 3rd AbSciCon where we've been co-sponsoring the meeting and feeding you coffee. So indirectly taking credit for all the amazing ideas that you guys are developing. Supporting the community has been incredibly important for Blue Marble Space, because the NAI has been instrumental, particularly on a personal story, in helping me develop my scientific growth, but also on the personal side, my personal growth. Scientifically, of course, I was lucky enough to come to the University of Washington and join their astrobiology program, although when I came to the University of Washington, it was for plasma physics in the aerospace engineering department. And then my first year of my master's, I took planetary atmospheres from David Catling, and then I had a quarter-life crisis. Here I was 6 years into engineering school and decided to start grad school all over again in Earth and Space Sciences and Astrobiology. And I start grad school in Earth and Space Science, and then David Catling gets this prestigious position in the UK. And I'm like, come on! He's supposed to be my wingman, but I was very lucky that David Montgomery and Roger Buick took me under their wing, and having Roger Buick as a graduate advisor is an incredible adventure, because the places he takes you to Western Australia and the stories he says really grabs you into the field. But on more important growth, I think, is a personal kind. I can't think of any other organization that has existed other than the NAI in their incredible support of the early career community. Particularly two important things. One is the AbGradCon conference and the summer schools. So I was actually this year is actually the 10-year anniversary of AbGradCon 2009, which was held in Seattle at the University of Washington. And I was the lead organizer among the grad student misfits for that conference. And little did I know that I working with friends, you could build something that's bigger than yourself. So really building this confidence of being the leading voice and believing in something that you're passionate about was something I could not have done without Carl's and NAI's vision of supporting grad students to organize a conference. Here's some funds, hang out, build community, build support. And I can say today, last week I was mid-career, but they changed the transition from 7-year to 10-year post-PhD. So now I qualify as an early career. So I really can say that now in my early career, my collaborators and best friends come from these bonds that we've made as grad students and through the summer schools. But also at AbGradCon in 2011, we wanted to kind of formalize our outreach, our community engagement efforts through SETI.org. I know maybe some of you have heard it, is our public engagement platform. But we didn't know how to be science communicators, how to support the next generation of scientists. We were, after all, early careers ourselves. And myself and some of our few co-founders, Sarah Walker, Betul Kessa, Grasshopper, Ilan Kuhn, and Julia Demirines, were like, well, we should go to a mentoring conference to learn what it's about to support, to learn the skills to mentor students who are interested, because astrobiology brings interest from all over the world. And so we asked Carl and NAI, hey, we would like to go to this mentoring conference in Albuquerque to help support our vision for SIGINET. And they were like, sure. And we're like, what? You go. And we did, and it's been an amazing experience in learning how to become a mentor. I think the growth of that has been the Young Scientist program that's been an offshoot of our Institute of Science. And now we are supporting 25 interns at NASA Ames who are through our program, where we bring in faculty from the Stanford School of Business to teach them how to give a presentation, and we give them a training in ethics and then in science, and then they build a community themselves as an offshoot of the mentoring skills that I was able to learn through the support of NAI. So I could talk for a long time about how NAI has changed my life fundamentally. I was going to be an engineer, and then starting a PhD looking at plasma physics and finishing it with looking at Precambrian rocks. It's kind of the story of all of us, I think. None of us have ever had a linear path in our career because of astrobiology, and that's the beauty of it. So we're excited to support to keep continuing supporting the community through our nonprofit activities, particularly at NASA Ames. And I encourage you to come visit our booth, number 2, downstairs, and poster number 17. So thank you.
My only complaint about the morning session was that Penny got all the hugs.
Still getting all the hugs.
Well, that's okay. Arsev, do you have something you want to show? Okay. This will disable that, so don't plan to use that except as a laser pointer, but otherwise we should now be able to get this in there. This was explained to me. Oh, yes, that's where we need to go. Okay. Great.
Hello, my name is Arsev. I'm the oddball. I'm a social scientist. I used to be an NPP at NAI Central between 2012 and 2014-15. I was brought in to study, actually, you. So you study everything else, I study you. My unit of analysis was, of course, the CAN teams, CAN 4, CAN 5, and CAN 6. But, I strayed. Astrobiology was so interesting. I studied concept of astrobiology, early career folks. There's a fly. And today, I would like to talk about my poster and the impact. My poster is about the impact of NAI in astrobiology. Of course, you can't see anything because this is quite small. I'm a social scientist, but I do qualitative research and quantitative research. And qualitative research is what your prejudices are. We are fly on the walls. I visited more than 20 labs all over the US for CAN teams, watched how people work together. I interviewed them. I interviewed 150 people. But I also looked at the publications and project reports you wrote and published and stuff. And I want to show you the first one. You can see on the right.
You have a laser. You have a laser right there, the small green button.
So you see the spike there for this red one? That's 1997, 1998. And that's the number of astrobiology and origins of life-related papers. So after the establishment of NAI, it skyrockets. Before that, it is 3, 5, at best 10 papers per year. But now we have close to 500 publications related to astrobiology and origins of life. That includes exoplanets as well. So NAI literally gave some structure. And in one of the interviews on top, which I am going to, I don't know, it's readable now? Oh, it's readable. Excellent. So this is an early career folk I interviewed some years ago. The names are not real, by the way. I changed it. So by Sanjoy's definition, that person is still early career, but actually he is or she is now mid-career. He says that NAI has done a really good job of getting a brand out there for astrobiology. Because as you already lived in it, as even 10 years ago, in some of the interviews, it was obvious that astrobiology, is it real? And I remember some students saying that their advisors trying to redirect them to more mainstream careers and stuff. Thank God they failed, and thank God we prevailed. So, and NAI really has given more of a pedigree and more of a reputation to astrobiology, which is really, really cool. The second thing I want to mention is, let's see, this one. Okay, you've seen this one, so this is bigger now. This is the first one I wanted to mention. This is about the interdisciplinarity of astrobiology. So this is a network map. These words come from the abstracts, titles, and the keywords of astrobiology and origins of life-related papers. So you're talking here about, geosciences, biology, astronomy. These disciplines are socially constructed. But these keywords are the real deal you're researching about. And you can, I showed it to astrobiologists, so it makes sense. I cannot fully interpret this because I'm a social scientist, but you can see the bigger domains, bigger subdomains within astrobiology here. It will tell you more. The yellow stuff, yellow cluster is about evolution, self-organization, emergence, or the blue stuff, astrochemistry, cosmochemistry, space dust, and things like that. So we can see where your research falls. We can identify potential collaboration opportunities that you haven't discovered yet. And the last one is the people. It's not all of you, because the resolution is really low in this one. There are, in NAIs, I did this for CAN 4 and CAN 5. I have 2 minutes, excellent. For CAN 4, CAN 5, there are close to 1,200 scientists, so you cannot see all the names here. And this is, names for 50 years and stuff, but you can see even Miller. Okay. Okay. But also, more, I don't want to say the term modern, but recent names, Jim Cleese or Chris McKay. And you can see, and each color represents actually a cluster. And the final thing I want to say about NAI is, getting back to this poster and these interviews, trying to enlarge. So NAI really, I've studied many communities. Astrobiology is not the only community I studied, but the attention given to early career folks here is very different than the other ones. Chemists have been trained the same way maybe for the last 2 centuries. Astrobiologists, one people actually mentioned that we are the misfits, people who do not fall into the traditional academic career paths. That is true. They take initiative, they do lots of stuff. But because you more senior folks are open-minded and provide them with opportunities and stuff, and you are doing your best to capture their inspiration, and they are reaching up to the plate. So in order to capture the next generation, we need not only to inform, as multidisciplinary seminars do. Back then, multidisciplinary was a buzzword. Now it's interdisciplinary. But also to inspire them to do the historic nature of the question that astrobiology poses. Great nations do great things, and NASA took it upon itself as a great agency of a great nation to embark on one of the most profound questions in human history. Astrobiologists dare to ask these big questions. That's a huge, huge difference compared to most of the other disciplines. And tackle that question in a scientific way, in a rigorous way, so it makes it a real science. So I cannot judge if NAI completed its mission or not, but I can say that NAI achieved its objectives, introducing an interdisciplinary science and creating a community that is sustainable. Thank you.
Thank you. Thank you, Arsev. And I think that it is very fitting that we finish this session with Ken Stedman, who worked very closely, as you are going to hear, with our founding director, Barry Blumberg, on a subject that is near and dear to both Ken and Barry's hearts. And so, Ken?
Let's see if we get this to work. Yeah. So basically, I'm the virus guy, except that unlike Barry, I'm not really a virologist. I just play one on TV, which is actually the CBC a few years ago, as you may hear about.
Barry was not a virologist either, and that was a point that he made repeatedly.
Yes. In fact, that is something that I have to agree with you on here. I don't think that any virologist is really a virologist. Sort of by definition, virology is interdisciplinary because you're thinking about structures, you're thinking about evolution, you're thinking about ecology, et cetera. So I think it's a really good match here. So how do I get this to actually work? This one.
I'm a Mac person. I never understand
Me too.
this. Arsev, we may need you back up here.
Yes.
Yeah, you got it.
Okay, here we are. So I also need to I think, thank ourselves to the goal that we actually have now as far as what I'm going to be talking about here for the next, 3.5 minutes I have left? That I didn't see a ball with viruses on it in your analysis. And so the next time in 10 years when all of these, next presentations come up, hopefully there'll be a really big blob in that particular aspect. But yeah, this is really, the virus focus group is really the brainchild of Barry Blumberg. And to hark back to some of the talks we had this morning, I wasn't a baby professor. I think I was actually in utero as far as professors were concerned when Barry first talked to me about this, because I was still a postdoc and just about to start my permanent position at Portland State University, where I've been ever since. So it's been a few years since then. But when a Nobel laureate asks you to be co-chair of a focus group, first your arm gets twisted a lot and you say, well, yeah, how high do I jump? But at the same time, realizing that, yes, I was co-chair, but basically I was going to have to do all the work. So what was that work? Basically, again, based on a lot of Barry's ideas, was just putting together workshops and meetings, particularly, again, going back to Barry not really being a virologist, getting out in the field. And that was really central to the way that Barry thought about doing science, even from a virologist's point of view. And what was the virus focus group supposed to do? Basically think about the origin and evolution of viruses, but also thinking about how viruses then are involved in evolution, in multicellularity, as we heard about a little bit earlier this afternoon. I think these are really, really critical kinds of aspects about what viruses do, what we know they do, and potentially could be involved in all kinds of other things. So my multidisciplinarity thing here at the bottom is about ecology, diversity, that there are beneficial viruses. I like to say viruses have a bad rap in many cases, and also thinking about viruses in the space environment. So again, we had a number of workshops. This is the very first one that we had was in Portland in 2003. Hopefully most of you recognize Barry in the middle. Some of you may see Sherry Cady hiding at the back here, and also hiding at the back in the room today. So I'll just going to quickly screen through some of these things. Then we had the first ever, as far as I know, astrovirology session at AbSciCon in 2004. And one of the things that was presented there by the late Roger Hendricks was that if you take all of the viruses on this planet and line them up end to end, they're about 200 million light years. Just ridiculous astronomical numbers of viruses, which we have on our planet. One place that we went to look, again with Barry, was Mono Lake, which is, as far as I'm concerned, like most environments, really understudied as far as viruses are concerned, and also was a very large group with multiple different participants, many students involved. As we also heard, the NII has been really wonderful about supporting students, supporting young researchers. This has also been published. Then we also had a field trip to my main stomping ground, which is in Lassen Volcanic National Park in Northern California. If you've seen that volcano here, Mount Rainier, if you keep going all the way south to the southernmost of the Cascade volcanoes, that's Mount Lassen, and has a wonderful area of all kinds of great geothermal areas, but also right next to that is the SETI Allen Array, and so we also visited there at the time. We have been very involved in trying to do public outreach, and as I mentioned here, I play a virologist on television. In fact, that last group where we were in Lassen, it was the Canadian Broadcasting Corporation that came with us, and I and a few real virologists were then on television with the CBC, and that's available. I've also been involved with the Astrobiology training program, Canadian, and then, as we saw before, also the California Academy of Sciences, looking for aliens on Earth. Are viruses aliens? I like that the viruses are actually really important for the development of all life rather than aliens per se. And then just wanted to finish up in terms of where the virus focus group has gone. One thing you have to do these days, if you ever end up on the cover of a journal, is to show the picture of that cover, because who looks at the actual printed journals anymore? So this was our review article in Astrobiology, beginning of last year, where we basically synthesized a lot of the things that have come out of those field trips and workshops and tried to put it into one semi-comprehensive, but of course we missed out on many things as well, as far as where we think astrovirology should go. And then that leads to where we have astrovirology today. There were a couple of posters, including this one, presented on Monday. Wednesday, there will be 4 more presentations with viruses. And we'll also have a couple of talks, both Wednesday and Thursday, on viruses. So please come and check those out. But most importantly is we need contributions from you in terms of how we can get the next time that we have a nice bubble plot and have viruses all over it. And that will be meeting tomorrow, 12:15 to 1:30 in the Balsam Room. We're setting up a workshop without walls. We don't have exact dates yet. We'll be in September, probably the week of September 16th. So stay tuned as far as all of that is concerned. Otherwise, you can come and find me. I will be heading to my poster, even though the poster session was yesterday. And with that, how many minutes do I have left?
Something close to 0.
Something close to 0. Well, this just leaves me to thank not only Barry and his memory as far as the founder of the NAI, but also some of the Director's Discretionary Fund that we got from Carl. Just as a quick spin-off, the funding we got from that turned into a patent which has now been issued in 5 different countries, the foundation of a company which I started about 5 years ago that may actually revolutionize getting vaccines to the developing world. So if you're more interested in that, come find me later. Thank you.
Thank you very much, Ken. I really, just in closing, would like to thank Ken, thank all the speakers, of course, but thank Ken for reminding us of the importance of a part of the biosphere that frequently gets ignored, or at least not given the respect that it deserves. And it was a part of the biosphere that was very dear to Barry Blumberg's heart. I'm sure most people in this room, but maybe not everybody, knows that Barry Blumberg, who was the founding director of the NASA Astrobiology Institute, had a long career in medicine. He was 72 years old when he became the NAI director, and he had won the Nobel Prize in 1976 for discovering the hepatitis B virus and developing the vaccine, which is still used today as the primary vaccine against hepatitis B. It's now made, it's manufactured in a different way than the way Barry made it, but it's the same vaccine. And if you don't know about Barry's life history and his story, I recommend that you read an article that appeared in Astrobiology, I forget how many years ago, maybe Sherry remembers, it was maybe 2015. At any rate, I wrote it, and I'm not recommending it because I wrote it. I'm recommending it so that if you don't know about Barry, you can learn about him. He was a remarkable human being. With that, I would like to thank my co-conveners, Penny and Dave. I would like to thank again all of the speakers, morning and afternoon, and especially thank all of you for spending your precious time here at AbSciCon with us in this session. Thanks.